Everyone, I am Dr. Shalini Maksane, Assistant Professor in Department of Biochemistry, St. GS Medical College and KM Hospital, Mumbai. In today's session, we will discuss about xenobiotics and biotransformation. Specific learning objectives of today's session are that you should be able to define the terms biotransformation, detoxification, and xenobiotics. You should be able to explain the process of detoxification with respect to its location in cell, organelle, enzyme, and coenzymes or cofactor involved in it. Describe a cytochrome P450 enzyme system with respect to detoxification process in human body. And you should be able to describe phase 1, 2 and 3 reactions involved in detoxification of the chemicals and drugs with suitable example. What is biotransformation? Biotransformation is the process in which a substance is changed from one chemical to another chemical by a chemical reaction within the body. In general, biotransformation reactions generate more polar metabolites that are readily excreted from the body. The liver plays the most important role in the biotransformation reactions. It can be beneficial as well as harmful sometimes. It can be divided into two types. First is detoxification and second is bioactivation. Detoxification. Detoxification is a biochemical process whereby the toxic substances are rendered less harmful and more water soluble. Lipophilic, non-polar and low molecular weight toxicants are converted into hydrophilic polar metabolite which can be easily excreted from the body. Compounds that are detoxified include compounds which are accidentally ingested like food additives, compounds which are produced in the body like ammonia converted into urea, bilirubin which is toxic to the brain is converted into conjugated water soluble bilirubin to be excreted into bile. Compounds produced by bacterial metabolism like amines produced by decarboxylation of amino acid due to bacterial metabolism are also included in this category. Now, second category is bioactivation. Bioactivation is the formation of harmful or highly reactive metabolic uh, compounds from non-toxic or relatively inert compounds. For some drugs, it is not the absorbed drug but a metabolite that has therapeutic action. But it is also known as toxication. The bioactive metabolites often interact with the body tissue to precipitate one or more forms of toxicity such as carcinogenesis, teratogenesis, and tissue necrosis. It can also cause direct toxicity, oncogene activation, and hypersensitivity reaction. For example, vinyl chloride is converted into vinyl chloride epoxide which damages DNA, RNA and can cause liver cancer. So bioactivation can be beneficial as well as harmful. What are xenobiotics? A xenobiotic is a compound that is foreign to our body. These compounds are either administered in our body in the form of various drugs or food additives, or we are exposed to them in the form of environmental pollutants, insecticides, etc. The principal classes of xenobiotics of medical relevance are drugs, chemical carcinogens, and various compounds that have found their way uh, into our environment by one route or another. More than 2 lakh manufactured environmental chemicals exist. Xenobiotics can be compounds which are produced by bacterial metabolism also. For example, amines which are produced by decarboxylation of amino acids by bacteria as I have already mentioned. Few examples of amines are histamine which is produced by histidine, cadaverine formed by lysine and tyramine produced by tyrosine or neurin produced by choline. Now we will discuss the role of cytochrome P450 enzyme systems in xenobiotic metabolism. The cytochrome P450 enzyme system is mainly required for hydroxylation reactions of xenobiotic metabolism. They are involved in the biotransformation reaction. They are like hemoglobin, they are hemoproteins. They are present in highest amount in liver and small intestine, but are probably present in all tissues. Mainly they are present in the membranes of the smooth endoplasmic reticulum. Cytochrome P450 are actually a superfamily of related heme-containing monooxygenase enzymes that participate in a broad variety of reactions. The name P450 reflects the absorbance uh, at 450 nm by the protein. Cytochrome P450s are so named because the enzyme was discovered when it was noted that preparation of microsomes that had been chemically reduced and then exposed to carbon monoxide exhibited a distinct peak at 450 nanometer. Among reasons that this enzyme is important is the fact that approximately 50% of the drugs human ingest are metabolized by isoforms of cytochrome P450. These enzymes also act on various carcinogens and pollutants. An extremely important function of the microsomal cytochrome P450 monooxygenase system found associated with the membranes of smooth endoplasmic reticulum is the detoxification of foreign compounds that are called xenobiotics. These include numerous drugs and various pollutants as petroleum products, pesticides, etc. The microsomal system can be used to hydroxylate these toxins using NADPH as a source of reducing equivalent. The purpose of this modification is twofold. First, it may itself activate or inactivate a drug, or second, it can make a toxic compound more soluble, thus facilitating its excretion in the urine or feces. In cytochrome P450 monooxygenase cycle, it required 2 electrons and 2 H ions for their catalytic cycle. As described in this diagram, you can see that monooxygenases incorporate one atom from molecular oxygen into a substrate to create a hydroxyl group. So here what happens, that hydroxyl group, this is the xenobiotic compound, and to add a hydroxyl group in that xenobiotic compound, it requires cytochrome P450 reductase enzyme, and it requires NADPH to as a electron donor. So what happens first, monooxygenases incorporate one atom from molecular oxygen into a substrate that is xenobiotic to create a hydroxyl group, with the other oxygen atom being reduced to water. NADPH provides the reducing equivalent which is required for by this series of reaction. As we know that it can transfer one electron at a time, electrons are transferred from NADPH to FMN and to FAD and FMN of cytochrome P450 reductase. Then it is transported to cytochrome P450. The ion of cytochrome P450 is the final acceptor. This leads to the reductive activation of molecular oxygen, and one atom of oxygen is subsequently inserted into the substrate. So here you can see that after cytochrome P450 accept electron, it causes activation of molecular oxygen. That oxygen get attached to cytochrome P450, which is attached to substrate, and then this lead to the reductive activation of molecular oxygen, and one atom of oxygen is subsequently inserted into the substrate. Now these 2 electrons along with 2 hydrogen ion react with another atom of oxygen to form water molecule. So here you can see that first oxygen atom is attached to cytochrome P450 and substrate complex, then it accepts another electron and 2H ion to form water molecule, and one oxygen is attached to the substrate to form hydroxyl group. While one of the atom of oxygen is incorporated into the substrate to form hydroxyl group, this dual fate of the oxygen accounts for the naming of monooxygenases as mixed function oxidases. So this enzyme is also known as mixed function oxidases because First, it causes hydroxylation of xenobiotic substrate, and second, it causes formation of water molecule. So here in this slide you can see the final reaction of hydroxylation of xenobiotic compound. So here you can see that this is the xenobiotic compound which is converted into less toxic compound by hydroxylation reaction by microsomal cytochrome P450 monooxygenase system, which causes addition of one oxygen atom into xenobiotic and one oxygen atom is incorporated into water molecule, and NADPH act as a electron donor for this overall reaction. So the for this hydroxylation reaction, what is required? First thing is cytochrome P450, which is a flavoprotein. Second thing is NADPH, which is required for reductive activation of molecular oxygen. Third thing is cytochrome P450 reductase enzyme. This enzyme is required for transfer of electrons from NADPH to cytochrome P450 via FMN and FAD. And finally, it requires molecular oxygen, which is required for hydroxylation of substrate molecule. Now, what are the cofactors which are required for cytochrome P450 mediated hydroxylation reaction? So, the cofactor required are NADPH, riboflavin, niacin. Niacin is required for formation of NADPH. Iron is also required, which is present in cytochrome P450 as it is a hemoprotein, which is iron is the final acceptor of electron from NADPH in cytochrome P450. And finally, it requires phospholipids. Lipids are also component of cytochrome P450 system. The preferred lipid is phosphatidylcholine, which is the major lipid found in membranes of endoplasmic reticulum. So this question may be asked in exam, that which cofactors are required for microsomal P450 mediated hydroxylation reactions of detoxification. So you should know what the name of these cofactors. Although hydroxylation reaction is the main reaction which requires cytochrome P450 enzyme system, but this system is also required for catalysis of wide range of reactions like deamination, dehalogenation, desulfuration, epoxidation, peroxygenation and reduction. But main reaction is hydroxylation reaction. Now, cytochrome P450 enzyme superfamily nomenclature. How these isoenzymes are named, we will discuss now. Around 150 isoforms of cytochrome P450 enzyme system has been discovered. Because of the large number of isoforms, it became important to have a systematic nomenclature for these isoforms of cytochrome P450. It is based on the structural homology. The abbreviated root symbol CYP, here you can see CYP, it denotes a cytochrome P450. This is followed by an Arabic number. So here you can see that Arabic number is 2. So this Arabic number designate the family. All cytochromes P450s which exhibit 40% or more sequence identity are included in the same family. That means all the cytochrome P450 which have 40% sequence homology will be included into family 2. Now this Arabic number is followed by a capital letter which indicate the subfamily. If two or more members exist, cytochrome P450s are in the same subfamily if they exhibit greater than 55% of sequence identity. The individual P450s are then arbitrarily assigned Arabic numerals. Thus, cytochrome, if in that example, we can say that cytochrome P450, that is a member of family 2 and subfamily C, is the ninth member of that subfamily. So this is how cytochrome P450 are named. The nomenclature for the gene encoding cytochrome P450 is same, which is used for the protein, but the only difference is that the symbol, if we are using it for the gene, it will be written in italics. These are some more characteristics of cytochrome P450 isoenzymes. Microsomal cytochrome P450 can act on both xenobiotics and endogenous compounds. They are less specific to substrate and can act on multiple substrates. Most isoforms of cytochrome P450 are inducible. Inducible means they can be activated. Substances that may cause overactivity of the cytochrome P450 enzymes are caffeine, alcohol, saturated fat, paint, fumes, and sulfonamides and barbiturates. There are many more also. So these compounds can activate cytochrome P450 enzyme. For example, administration of phenobarbital causes hypertrophy of smooth endoplasmic reticulum and a 3 to 4 fold increase in the amount of cytochrome P450 within 4 to 5 days. The mechanism of induction has been studied extensively, and in most cases, increased transcription of mRNA for cytochrome P450 was observed. However, in certain cases, the induction involved stabilization of mRNA and enzyme stabilization. Another important fact about phenobarbital induced activation of cytochrome P450 is that phenobarbital induces cytochrome P450 enzyme, which is the same enzyme which metabolizes warfarin. If a patient is taking warfarin, is given phenobarbital, the enzyme activity of cytochrome P450 will increase and warfarin will be catabolized more. So it breaks down quickly and dose become inadequate. Similarly, ethanol induces cytochrome P450 type family 2 and E1 subfamily E and first number of this enzyme which metabolizes many carcinogens. Thus, the risk of carcinogenicity is increased after the use of ethanol because ethanol induces the activity of this enzyme which metabolizes carcinogens. Another characteristic of cytochrome P450 enzyme is that they exist in polymorphic forms, means they have genetic isoforms, some of which have low catalytic activity while other have high catalytic activity. This is the explanation for the variation in drug responses noted among many patients. Though cytochrome P450 enzymes are mainly present in endoplasmic reticulum, but mitochondrial cytochrome P450 also exists. They uses NADPH linked flavoprotein called adrenodoxin reductase and a non-heme ion sulfur protein adrenodoxin. In addition, the specific cytochrome P450 isoforms involved in steroid biosynthesis are generally much more restricted in their substrate specificity. Means mitochondrial cytochrome P450 enzymes are more specific for their substrate, while microsomal cytochrome P450 enzymes are less specific. They can act on multiple substrates, while mitochondrial one have can act on specific substrates. Now, phases of detoxification. Detoxification reactions are categorized depending on the normal sequence with which they tend to react with a xenobiotics as phase 1, 2 and 3. Mainly there are phase 1 and phase 2 reactions. Phase 3 reactions are less common. Phase 1 reactions involve the creation of a functional group in the substrate molecule or the transformation of an existing functional group. Alteration of xenobiotic molecule by adding a functional group in phase 1 can cause detoxification or intoxication. When the phase 1 pathway converts a toxic chemical into a less harmful chemical, this is achieved by various chemical reactions such as oxidation, reduction and hydrolysis. During this process, free radicals are produced, which if excessive can damage the liver cells. Antioxidants such as vitamin C, E and carotenoids reduces the damage caused by these free radicals. If antioxidants are lacking and toxin exposure is high, toxic chemicals become more dangerous. Some compounds may be converted from relatively harmless substances into potentially carcinogenic substances, which is called intoxication. Excessive amount of toxic chemicals such as pesticides can disrupt the cytochrome P450 enzyme system by causing overactivity or what is called induction. This will result in high levels of damaging free radicals being produced. The danger is if their reactive molecules are not further metabolized by phase 2 reactions, they may cause damage to proteins, DNA, RNA within the cell. Phase 2 reactions are conjugation reactions. In most cases, the conjugation will make the compound non-toxic and easily excretable. Conjugation with a conjugating agent convert lipophilic compound to hydrophilic compound. Main conjugating agents are glucuronic acid, sulfate, cysteine, acetic acid, glycine and glutamine, which we will discuss in coming slide one by one. Phase 1 reaction products are the substrate for phase 2 reactions. The major enzymes involved in these cases are uridine 5-diphosphate glucuronil transferase, glutathione S transferase and sulfotransferase. Depending upon the nature of reactions, phase 1 reactions are categorized into first oxidation reaction, second reduction and third is hydrolysis. In phase 1 reaction, the majority of such metabolic reactions are oxidation reaction. Most of the phase 1 reactions are oxidation reaction, but numerous reactions of reduction are also known. In oxidation reaction, transfer of one oxygen atom or two oxygen atom can be done. It can be an aliphatic or aromatic depending upon the type of substrate, as I have discussed during cytochrome P450 action, where oxidation take place and there is addition of one oxygen atom. And oxygen group can be added to sulfur or nitrogen group also, and it can be epoxidation also. So in oxidation, self-oxidation, N-oxidation, epoxidation and hydroxylation can take place. Reduction reaction involves one or two electron transfer, and third class is hydrolysis. Here the degree of oxidation of substrate remain unchanged. Water molecule is used to break xenobiotic substrate. Now this is the example of hydroxylation phase 1 reaction. Here toluene is hydroxylated to benzyl alcohol by mixed function oxidase, or which is called monooxygenase system. Here you can see one oxygen atom is added, NADPH is converted into NADP and hydroxylation of toluene is done, and in that reaction one water molecule is also formed. Now this is another example of hydroxylation, but sometimes both phase 1 and phase 2 reactions are required for complete detoxification. For example, the conversion of benzene to phenol. Here you can see that benzene is biotransformed initially into phenol by a phase 1 oxidation reaction, where hydroxyl group is introduced into the benzene to form phenol. Now phenol has a functional hydroxyl group that is then conjugated by phase 2 reaction. So it can be conjugated by sulphation reaction or by addition of glucuronic acid. So there are two fates for phase 2 reaction of phenol. Phenol is converted into phenyl sulfate or it can be converted into phenylglucuronide by glucuronic acid. Now, oxidation of alcohol groups. This is also a type of detoxification reaction. It occurs in liver. The oxidation and detoxification of alcohol is an important function of liver. Two enzymes are involved in this process. First is alcohol dehydrogenase, which is a cytosolic enzyme. This converts ethyl alcohol into acetaldehyde, and the removed H ions are accepted by NAD+. Another enzyme is aldehyde dehydrogenase. It is a mitochondrial enzyme. It is also NAD-linked enzyme. It converts acetaldehyde to acetic acid by oxidation. So this is how the detoxification of alcohol take place in liver. Another example of oxidation of alcohol group is conversion of benzaldehyde to benzoic acid. Here you can see in this diagram. Here also NAD is it act as a hydrogen acceptor for oxidation of benzaldehyde. Oxidation of some compound may result in production of substances which are more toxic. In previous slide you saw that oxidation decrease the toxicity of alcohol, while here the oxidation increases the toxicity of some alcoholic compound like conversion of methanol to formic acid and conversion of ethylene glycol to oxalic acid. They are here the toxicity is increased by oxidation. Another type of phase 1 reaction is reduction reaction. Some of the reductases also contain cytochrome P450 and are flavoproteins in nature. The major group of compounds which are reduced and detoxified by the liver are nitro compounds. These are reduced to their amines, while aldehyde and ketone are reduced to alcohols. An example for reduction is conversion of nitrobenzene to aniline. The enzyme responsible for reduction of this xenobiotic require NADPH as a cofactor, which provide H ion for reduction. The other substrates for reductive reactions include epoxide, heterocyclic compounds, and halogenated hydrocarbons. Another type of phase 1 reaction is hydrolysis. Hydrolysis is a chemical reaction in which the addition of water split the toxicant into two fragments which are smaller molecule. The hydroxyl group is added into one fragment and hydrogen atom is incorporated into another part. Esters, amines, hydrazines, amides, glycosidic bonds, and carbamate are generally biotransformed by hydrolysis reaction. For example, aspirin and acetanilide, procaine, xylocaine, aliphatic esters, they are all biotransformed by hydrolysis reaction. So here you can see that acetanilide is hydrolyzed into aniline and acetic acid by adding water molecule. Here you can see that one hydrogen molecule is added into one fragment and hydroxyl group is added into another fragment of acetanilide to form aniline and acetic acid. In another example, you can see that aspirin is hydrolyzed into salicylic acid and acetic acid by addition of water molecule. The hydrolytic reactions, contrary to oxidative or reductive reactions, do not involve change in the state of oxidation of the substrate, but involve the cleavage of substrate molecule by taking up a molecule of water. Here the oxidation state is not changed of the substrate, but it is broken down into pieces, smaller pieces by addition of water molecule. Now we will discuss phase 2 reactions. First is sulphation reaction. Sulphation of xenobiotic decreases the toxicity. The higher polar sulfate conjugates are readily excreted through urine. Often glucuronidation or sulfation can conjugate the same xenobiotic as we have discussed in earlier slide, where phenol is converted into phenyl sulfate and phenyl glucuronide, both type of compound. Phenolic and alcoholic compounds are conjugated with sulfate. The enzyme which is required for the reaction is sulfotransferase, and the sulfate group is transferred from phosphoadenosyl phosphosulfate. The conjugation of sulfate are called the sulfate ester conjugates, or they are called ethereal sulfates. Functional group capable of forming sulfate conjugates include phenol, alcohol, arylamines, N-hydroxylamines, and N-hydroxyamides. Drugs undergoing sulfate conjugation include chloramphenicol, phenols, and adrenal and sex steroids. They all can be sulfated. So here in this reaction you can see that sulfation of phenol causes formation of phenyl sulfate in the presence of sulfotransferase enzyme. Indol is converted into indoxyl sulfate, and sulfate donor is phosphoadenosyl phosphosulfate. Another type of phase 2 reactions are glucuronidation. Conjugation with glucuronic acid is the most common and most important phase 2 reaction. The biochemical donor of glucuronic acid is uridine diphosphate glucuronic acid, and the conjugating agent is glucuronic acid. The enzyme which carried out this reaction is called uridine diphosphate glucuronyl transferase. It is present in microsomes of most tissues, but liver is the most active site of glucuronide synthesis. Various drugs also undergo glucuronidation, including morphine and paracetamol. Certain endogenous substances such as steroid, bilirubin, catechols, and thyroxine also form glucuronides. Occasionally, some glucuronides conjugates that are excreted in bile undergo deconjugation process in the intestine, which is mediated by enzyme beta-glucuronidase. This releases a free and active drug in the intestine, which may be reabsorbed and undergo enterohepatic circulation. This slide depicts the conjugation of bilirubin. Here you can see that UDP glucuronic acid react with bilirubin in the presence of enzyme glucuronyl transferase and form bilirubin monoglucuronide. And further, bilirubin monoglucuronide again react with UDP glucuronic acid to form bilirubin diglucuronide, which is known as conjugated bilirubin, which is excreted in bile, and it is soluble in blood plasma. Other example of glucuronidation are presented in this table. You can see first compound is phenol, which is converted into phenyl glucuronide, and here the glucuronic acid is attached to the phenol by O-glucuronide linkage or glucosidic linkage. Another example is benzoic acid, which is converted into glucuronic acid monobenzoate, and the type of bond which is formed is ester. Third is steroids compound, which are converted into glucuronide or steroid, and the glucuronide is attached to steroids with hydroxyl group. Last example is amines. They are converted into N-glucuronide, and glucuronide is attached to amines by amide linkage. Now phase 2 reactions where conjugation occur with glutathione and cysteine. As you know, glutathione is a tripeptide which is consisting of glutamic acid, cysteine and glycine. Glutathione is commonly abbreviated as GSH because of the active group sulfidyl group which is present in glutathione in cysteine. Electrophilic xenobiotics such as certain carcinogens and drugs are conjugated to the nucleophilic glutathione. The interaction between the substrate and glutathione is catalyzed by enzyme glutathione transferase in liver cytosol. Alkyl or aryl halides, epoxides which are formed in phase 1 reactions, and alkenes are detoxified in this manner. if the potentially toxic xenobiotics are not conjugated to glutathione, they would be free to combine covalently with DNA, RNA and protein of cell, which can cause serious cell damage. That's why glutathione is important defense mechanism against these toxic compounds. This glutathione conjugate, this is either directly excreted in bile or it is further metabolized to form mercuric acid. The glutamyl and glycine groups which belong to glutathione are removed by specific enzyme gamma glutamyl transpeptidase and peptidase. So from glutathione, the cysteine glycine group, the glutamyl and glycine group are removed from this glutathione with the help of enzyme GGT and peptidase. Now cysteine group remains on the substrate, xenobiotic substrate. This resulted in the formation of R-cysteine. This R-cysteine reacts with acetyl coenzyme A in the presence of an acetyl transferase enzyme which transfers this acetyl group to cysteine attached xenobiotic moiety to form mercuric acid. This mercuric acid is now excreted in urine. Conjugation with glutathione and mercuric acid formation is a minor but important metabolic pathway in animals. The cysteine, which is also derived from glutathione, it is also an active conjugating agent. Cysteine can directly acetylate first and then acetylcysteine is formed which get conjugated with toxic substances like bromobenzene, chlorobenzene, benzyl chloride, etc. Here also the end product is mercuric acid, but here acetylation is done by acetic acid in these types of reaction. For example, bromobenzene react with cysteine and acetic acid to form bromophenyl mercuric acid which is excreted in urine. Acetylation reactions of phase 2. Conjugation with acetyl group is an important metabolic pathway for drugs containing the amino groups. Here you can see this is amino group. The cofactor for these reaction is acetyl coenzyme A. The acetyl group is donated by acetyl coenzyme A. And the enzymes are non-microsomal and acetyl transferase which are located in the soluble fraction of cell in various tissues but mainly in liver. Acetylation is the primary route for biotransformation of sulfonamide and isoniazid drugs. These drugs undergo acetylation before their excretion. Polymorphic types of acetyl transferase enzyme exist, resulting in individuals who are classified as slow or fast acetylators. It influences the rate of clearance of drugs such as isoniazid from blood. Slow acetylators are more subjected to certain toxic effects of isoniazid because this is cleared slowly in these types of individuals. So here you can see the acetylation reactions of isoniazid and sulfonamide drugs, and in the presence of enzyme and acetyl transferase, acetyl group is donated by acetyl coenzyme A. So isoniazid is converted into acetylated isoniazid and sulfonamide is converted into acetyl sulfonamide, which are now then excreted from the liver. Methylation reactions. A few xenobiotics are subjected to methylation for their excretion. The reaction is catalyzed by enzyme methyltransferase and S-adenosyl methionine act as a methyl donor. Conjugation with methyl group involves transfer of a methyl group from the cofactors S-adenosyl methionine to the acceptor substrate. So S-adenosyl methionine act as a methyl donor which get attached to the xenobiotic substrate compound. For example, epinephrine and non-epinephrine are methylated before their excretion. Catechol O methyltransferase converts epinephrine to metanephrine, and pyridine is excreted as N-methylpyridine Mercaptoethanol is excreted as 5-methyl-mercaptoethanol. Conjugation with amino acids. Glycine and glutamine. Uh, Many aromatic carboxylic acid are conjugated with these glycine and glutamine. Glycine can act on cholic acid to form glycolic acid. Glycine can act on benzoic acid to form hippuric acid. Glutamine also act as a conjugating agent. For example, phenyl-lactic acid react with glutamine to form phenylacetyl glutamate, which is now excreted. Now, conjugation with thiosulfate. Conjugation with thiosulfate is an important reaction in the detoxification of cyanide. Conjugation of cyanide ion involves transfer of sulfur atom from thiosulfate to the cyanide ion in the presence of enzyme rhodinase to form inactive thiocyanate. Thiocyanate formed is much less toxic than the cyanide and it is excreted in urine. Sodium thiosulfate is used for the treatment of acute cyanide poisoning. Administration of a combination of sodium thiosulfate and sodium nitrite is given to the patient by intravenous infusion. Sodium thiosulfate exerts its antidotal effect by serving as a sulfur donor. It increases the rate of rhodinase enzyme which catalyzes biotransformation of cyanide to thiocyanate, thus decreases its toxicity. Now, what is the clinical significance of all these xenobiotic metabolism? The knowledge of xenobiotic metabolism is important to understand pharmacology, pharmacogenetics, therapeutics, toxicology, management of cancer, and drug addiction. All these areas involves administration of or exposure to xenobiotics. We are exposed to various xenobiotics in environment. When the xenobiotic is a drug, phase 1 reactions may produce either its active form or may diminish or terminate its action. If it is a pharmacologically active agent in the body without prior metabolism, there is no role of there is no role of xenobiotic. The diverse effects produced by drugs comprise the area of study of pharmacology. So, by understanding the metabolism of xenobiotics, we can know how various drugs act in our body, how they are either they are activated or they act directly, or after administration whether their toxic effects increases. So all these things can be understood by having the knowledge of xenobiotic metabolism. Certain xenobiotics are very toxic even at low levels like cyanide, while few xenobiotics, including drugs, they do not exert toxic effect if sufficient amount of these drugs are administered. The toxic effects of xenobiotics are that they can cause cell injury, which can be severe enough to result in cell death. The active metabolite of xenobiotics bind covalently to cell membrane, macromolecules like DNA, RNA, and protein. The reactive species of a xenobiotic may also bind to a protein, which can cause change in antigenicity of those proteins. The xenobiotic is said to act as a heptane, that is, small molecule that by itself does not stimulate antibody synthesis, but will combine with antibody once formed. The resulting antibodies can then damage the cell by several immunological mechanisms that disturb normal cellular biochemical processes. Now, the reactions of activated species of chemical carcinogens with DNA are thought to be of great importance in chemical carcinogenesis. If we know how a carcinogen react with our cellular component, we can target specific drugs according to the mechanism of action of that carcinogenic compound. So, understanding of how pro-carcinogen is converted into carcinogen, and how this carcinogen react with our cellular component. All these knowledge which comes under metabolism of xenobiotic are important for management of cancer. Some chemicals like benzopyrene require activation by monooxygenases in the endoplasmic reticulum to become carcinogen. So they are called indirect carcinogens. And next is gene polymorphism, which we have already discussed, means which says that certain cytochrome P450 exist in polymorphic forms, some of which exhibit low catalytic activity, and these observations are the important explanation of the variation in drug responses noted among many patients. Means if a particular drug is acting on one person but it's not acting on another person, it is explained by the gene polymorphism of cytochrome P450 enzyme. So accordingly the treatment can be given to the patient. So these are the clinical significances of xenobiotic metabolism. Thank you very much for listening this video.